Good morning or good afternoon, everyone. Today, I would like to take this opportunity to introduce you a new method for monoclonal antibody by single B-cell express cloning. I especially want to share with you our experience of using transgenic animals and the special animal strains. I will show you when the special animals used together with the single B-cell cloning method, we are able to develop antibodies more rapidly, more convenient, and we were able to generate antibodies with higher quality. So since the first monoclonal antibody was generated in the 1970s, a lot of methods have been developed for developing monoclonal antibodies. The first thing I want to point out is that although now a lot of in vitro methods have been developed, but today animal immunization remains the dominant source for antibody, whether it's used for research or clinical diagnosis or therapeutics. Most antibodies were still derived from animal immunization and the B cells generated from the animals. From there, eventually got the antibodies either in its conventional form or single domain antibodies, which are probably better known as nanobodies that are being increasingly used today. In this slide, I would first summarize a few methods that are commonly used today for monoclonal antibody development. First method is the classical hybridoma method. In this method, the B cells from the animals were first fused with myeloma cells to get the hybridoma cells, which are immortalized and able to secrete antibodies. The disadvantage of this method is that it is quite slow and usually takes many months to complete. Another disadvantage is that the original clone isolated from this method are usually not pure and require several rounds of subcloning to eventually get a pure clone. The hybridoma cells can then be used for large-scale antibody production, which is an advantage. But sometimes the cells can be unstable and eventually lose antibody secretion. The second method I would like to introduce is also a Nobel Prize winning technique. It's called surface display. Most often used fidget display and yeast display. This method is also slow and it's kind of unnatural. By unnatural, I mean that first, the antibodies are expressed in an environment that is quite different from the mammalian cell environment where the antibodies were originally from. The other unnatural thing is that in surface display, it is very difficult to keep the original native heavy and light chain pairs. In most approach, people just amplify the heavy chain and the light chain DNA separately and randomly combine them. The random combination of heavy and light chain will affect the antibody quality for sure. The third method, which I developed more recently, is the single B-cell sequencing method. Today, due to the rapid development of next-generation sequencing technique, single cell barcoding and sequencing can be routinely carried out in a regular lab. A number of companies now offer fully automated single B-cell barcoding and sequencing solutions. According to the data provided, thousands or even more antibody sequences can be obtained in just a few days. But this sequence still need to be synthesized and cloned into an expression vector to finally produce the antibodies for screening. So in a sense, it's still lab intensive. Also, it usually requires a special instrument, which can be quite expensive. The last method I wanted to mention is the single B-cell culture method. Due to the benefit from the many years study of B-cell biology, several cytokines has been identified that can sustain single B-cell survival and amplification when cultured in the well. The cultured cells will secrete antibodies into the wells and eventually build up to enough concentration that can be used for screening. The single B-cell culture is successful, however, it's relatively technically demanding. It usually requires a lot of optimizations to be able to carry it out reliably. Another disadvantage is that the antibody production in this method is usually low. For each well, you typically get only 100 nanogram or even less. That is, although enough for screening such like ELISA, but often not enough for other assays. To enable fast antibody discovery and reduce the workload, we developed a new method which we call SBEAD, stands for Single B-cell Express Antibody Discovery. In SBEAD, the B cells from the immunized animals are sorted by FACS. Then mRNA was extracted from the sorted single cells and quickly amplified through a specific procedure, which produce materials that can be used directly for cell transfection. In 5 days, by transfecting 293 cells or CHO cells, we were able to get enough antibodies, either in conventional form or single domain antibody format for screening. SBEAD is a species-independent method. We have successfully applied it on mouse, rabbit, alpacas, or even human cells. But we do focus on 2 species and work much less on others. The first species is rabbit. Rabbit is actually the most classical antibody animal. It was used specifically for antibody production in 1815. That's more than 100 years ago. The reason we choose rabbit is that it is known to produce high affinity antibodies. According to many literature reports, especially the studies from Alpacas, rabbit monoclonal antibodies typically have 10 to 100 times higher affinity than mouse monoclonal antibodies. Rabbits do have a strong immune system, which is ideal for small molecules or heptans that sometimes do not even trigger immune response in mice, but it can work well in rabbits. And another advantage is that rabbit antibodies can often react with both human and mouse antigens, making it ideal, especially if you are developing therapeutic antibodies, because today most of the disease models are mouse models. By having an antibody that can react with both the human and mouse target, you can easily carry out preclinical study in mouse first, then translate it to human studies. The last reason is that I will show you in the later slides that in the past we have developed many special rabbit strains and transgenic rabbit lines that can be used conveniently for antibody development. The second species we focus on is alpaca. Many of you know that alpaca and other camelid animals produce a quite different antibody from other mammals in that they consist of only heavy chain. By having only one chain, the antigen binding domain can be made very small into a single domain antibody, or otherwise known as nanobodies, which has a size of only 1/10 of a conventional antibody. Due to the small size and single chain, the antibodies can be used very flexibly, and you can easily create bi or even tri-specific antibodies from them. These nanobodies can be expressed not only in mammalian cells, but also in coli or in yeast, saving a lot of cost. Also, nanobodies can find many applications that can not easily be done by conventional antibodies. For example, they can be expressed in the reducing intracellular environment to bind the cytoplasmic antigens. Due to these specific properties, we focus our method on rabbit and alpacas. And I will show you a few examples in the later slides. Compared to the existing method that I show you in the first slide, SBEAD has many advantages. Specifically, when compared to hybridoma method, it is much faster. With SBEAD, you can develop monoclonal antibodies in as short as 11 weeks. while if you are using hybridoma, it usually takes more than 25 weeks. Since the antibodies were developed from sorted single cells, the monoclonality is guaranteed. And with the determined sequence, you can produce recombinant antibodies unlimitedly in your own lab. You don't have to worry about the stability of the cell lines. Compared to the surface display method, speed is also faster. Surface display method usually takes more than 21 weeks, and it's more natural because everything is done in the mammalian expression system, and the native heavy and light chain pairs are kept in the method. Compared to the single B cell sequencing, speed is also faster. It takes about 1 week less, and it does not require a special automated instrument, so it's less costly. It's actually also less labor intensive because it does not involve the cloning of many synthesized DNA into the expression vector. Compared to the single B cell culture method, speed is also faster. Another advantage is that it produces more antibodies for screening. Like I mentioned, in single B cell culture, you usually can only produce about 100 nanogram antibody each well for screening, but using amplified RNA transfection, we typically can produce more than 1 microgram antibody in each well of 96 well plate. I will show you several examples of speed antibody development in rabbits and alpacas. We have done a lot of antibody development in regular animals, but today I would like to show you a few examples of using speed together with the transgenic animals or special animal strains that we developed during the many years of research in the past. The first animal I would show you is LucAb animals, which produce antibodies fused with the luciferase reporter. The idea is that by knocking in a luciferase reporter gene into the overriding frame of the antibody gene, the antibody produced will naturally carry endogenous luciferase reporter so that it can be detected without the using of a secondary antibody. So that the antibody screening can be carried out faster, more conveniently, and often more accurate. Turned out it's not so easy to generate such animals. We actually failed several times until we finally were able to get healthy animals producing strong luminescent antibodies with an immune system that is as robust as actually indistinguishable from the wild-type animals. We generated both mice and rabbits. The test result showed that the animals produced strong luminescent antibodies in the serum that can be immobilized by protein G. The LucAb, because it naturally carries the luciferase reporter, it can be used in many assays without the using of a secondary antibody. By bypassing the secondary antibody, the assays can be carried out faster, and it involves less experimental steps to reduce experimental mistakes, especially when the lab staff needs to handle a lot of antibody samples. This is especially helpful. A typical ELISA assay using the LucAb can be completed in less than 40 minutes with only one washing step. A typical Western blot using the LucAb takes only less than 1 hour, again using only one washing step. The result show here that the LucAb luciferase can detect antigen as low as 1 nanogram. The sensitivity is probably not as high as the best regular Western blot, but is enough for most antibody screening assays, especially during the antibody discovery stage. Another big advantage of the LucAb is that the antibody luminescence signal is almost in perfectly linear correlation with the antibody concentration. This made the quantification of the antibody concentration very easy. By adding substrate, you can quickly quantify each antibody concentration, and the concentration information often can help interpret the screening assay result more accurately. For example, when a typical 9-6 well plate screen ELISA assay was carried out, the assay identified one well with the strongest signal. Usually, this well will be selected as a candidate and moved to the next stage. By adding back the medium, the same amount of medium used in the ELISA, we can and read the plate again. You can quickly measure how many antibodies were originally added to the ELISA assay, and then the ELISA assay can then be normalized to get the new result, and the normalized result showed another well, different well, showing the strongest binding, which should correlate to the antibody affinity more accurately. We have used the LucAb animal to develop many antibodies with high affinity. In an example shown here, we have used it to develop antibodies against two COVID-19 antigen proteins, including the nucleocapsid antibody, nucleocapsid protein, and the RBD domain of the spike protein. For both antigens, because we due to the luciferase signal of the LucAb, we were able to screen a large panel of antibody pairs conveniently, and were able to finally identify antibody pairs that were able to detect as low as 0.5 picogram per ml COVID-19 antigen protein in biological samples. The result show here that for both nucleocapsid and RBD, our antibody pair were able to differentiate biological samples that contain as low as 0.5 picogram per ml antigen from the negative control samples. By having a low detection limit of 0.5 picogram per ml, our antibody pairs are actually much more sensitive than other ELISA kits from the other vendors. The other special animal I want to show you is a rabbit called a Hylmmune rabbit. Hylmmune rabbit is a strain that have increased B cell population. Our test showed that with the same immunization, Hylmmune rabbits typically produce 3 times antigen-specific B cells than regular New Zealand white rabbit. By having more B cells, the single B cell cloning method, such like SBEAD, can be carried out with high successful rate. For example, by using the Hylmmune rabbit and the SBEAD method, we were able to develop antibodies against many antigens that were typically difficult to generate antibody, including antigens of small molecule compounds. In an example shown here, the Hylmmune rabbits were immunized with a small molecule compound fentanyl, which has a molecular weight of about 300. The result showed that the immunization generated a strong immune response in the Hylmmune rabbits. When the B cells were isolated and the SBEAD were used to develop antibodies, an antibody was discovered that can detect the fentanyl with a high sensitivity of an I50 value as low as 0.02 nanogram per ml. For comparison, the commercial ELISA kit used in the forensic lab has an I50 of about 0.3 nanogram per ml. That means our antibody is about 10 times more sensitive than the commercial antibody. Also, interestingly, our antibody can also detect carfentanil, which is an important derivative of fentanyl molecule. No other commercial antibodies can detect both molecules. Our antibody can detect the carfentanil with an R50 as low as 0.06 nanogram per ml. Commercial ELISA kit for carfentanil detection has to use a separate antibody with an detection limit of about 0.1 nanogram per ml, which is less sensitive than the antibodies that we developed. The high immune rapid, as well as the animals that I will show later, does not have luciferase fusion, so that is and concentration cannot be measured so conveniently. Because we have noticed that by knowing the antibody concentration, the screening result can be interpreted more accurately. So we tested a few various methods for antibody quantification, and we are now using the ABC-assay method in our lab. ABC-assay is a fast mix and read method, which involves very simple procedure and takes only less than 5 minutes to complete. Our test showed that the method can be used for mouse, human, and rabbit antibody quantification. And it is especially sensitive for rapid IgG quantification with a low detection limit of about 1 nanogram per ml and a linear range of about 4 log. In antibody development, the most time spent is actually on animal immunization. For example, in SBEAD, it takes only less than 2 weeks to clone the antibodies to finally get the recombinant antibody protein. But the animal immunization process takes 2 to 3 months. To test if the high number of high B cell number of the high immune rapid can be used to develop antibodies faster, we tested the high immune rapid immunizing with carcinoembryonic antigen protein immunization. The result showed that after only 4 weeks of immunization, the animal already generated a significant amount of antibodies in the serum. Taking advantage of the high number of B cells from the high immune rabbits, we used the SBEAD method to develop antibodies against the antigen CEA. And our result showed that out of the 88 B cells, single B cells that were sorted, we were able to develop 57 CEA binding antibodies with various affinities. Some of the high affinity antibodies can reach the KD value of picomolar units. There are other antigens that are weakly immunogenic. For example, when we attempted to develop an antibody against the A-beta-42 peptide, when the high immune rabbits were immunized with the unconjugated A-beta-42 peptide, even after full immunization cycle, the animal still only generated very, very weak immune response, which is about the serum titer result showed that it is about more than 10 times weaker than normal response. Because of the high number of B cells we can obtain, we were still able to get many monoclonal antibodies from this immunized animal. The result showed that out of the 88 single B cells sorted, we were able to obtain 30 A-beta-42 binding antibodies, and many of them have picomolar affinity, binding affinity. The last rabbit strain I want to show you is some rabbit which we call Huab rabbit. Today, antibodies are used as a very important therapeutic reagent. Among all the antibody drugs that received the FDA approval, most of them are actually originally developed from animal first and then humanized. Of these humanized antibodies, they are almost exclusively from mouse in the past. Until recently, things started to change. In 2019, 2 humanized rabbit antibodies received FDA approval. The reason why rabbit antibodies are used much less often for therapeutics than mouse. There are many reasons for that. But one of the reasons is that the rapid antibody has, especially the light chain, has a structure that is quite different from human antibodies. The most antibodies from regular rapid in the light chain, there is a special disulfide bond connecting the variable domain and the constant domain. But this special disulfide bond does not exist in human antibody. When the rapid antibody is humanized, this special disulfide bond will be lost, so that this often leads to the disruption of the rapid antibody structure and the loss of antigen binding affinity. To solve this problem, we identified this Huab rapid string. Our test showed that most antibodies from this special string does not have the special disulfide bond, which makes the antibodies from this special string more like human antibodies and can be more easily humanized. In an example, when we try to develop the antibody against the VEGF-A, we were first able to develop a total of 150 monoclonal antibodies. Out of the 150 antibodies, we selected 43 that have affinity of KD less than 1 nanomolar. From the 43 high affinity antibodies, we identified 11 of them which are able to inhibit the cell proliferation function of VEGF-A. And we made the chimeric antibodies from all the 11 antibodies by substituting the constant region with the human constant regions. Normal rapid antibodies, by such a chimerization, will lose the disulfide bond and lose the antigen binding affinity. But all the 11 antibodies developed from the Huab rapid retained high binding affinity to VEGF-A, which is about 10 times higher than Avastin, which is commonly used VEGF antibody used in therapeutics. We choose 3 of the antibodies to further humanize the variable domain, and finally get the 3 humanized monoclonal antibodies. The result showed that all the 3 antibodies retained biological activity that can completely inhibit the function of VEGF-A at a concentration less than 1 nanomolar, while for Avastin, it requires about 7 nanomolar to achieve the same complete inhibition. I would like to use the last 2 slides to show you examples of SBEAD application in a different animal, which is alpaca. Alpacas are now frequently used for single domain antibody development. Phage display or yeast display are almost the dominant method used for such applications. We started to test if the single B-cell method SBEAD can used to derive single domain antibodies more rapidly. For this purpose, we first derived an antibody that can specifically bind to the VHH antibodies, but not the VH antibodies. By staining the alpaca B cells, either from PBMC or spleen with this VHH specific antibody, we were able to successfully enrich the B cells that express IgG2 or 3, but not IgG1. IgG2 or 3 are the antibodies with heavy chain only. IgG1 is the conventional antibody. The PCR result showed that the enriched B cells almost exclusively express IgG2 or 3 with very little IgG1 expressed. When the IgG2, 3 enriched B cells were further sorted by antigen binding, we were able to develop single domain antibodies binding to the PD-L1 antigen with an affinity of about in the nanomolar range. When the cells from the same animal were used for single domain antibody development by yeast display, we were also able to obtain single domain antibodies with about the same affinity. But in each display, because library has first to be constructed and it has to be enriched by several rounds of enrichment. It takes more than 6 weeks to finally get the antibodies. With SBEAD, we were able to get the same affinity of single domain antibodies in less than 2 weeks. By showing these examples, I want to give a summary here. By showing these examples, I want to show you that SBEAD is a novel monoclonal antibody development method which is faster than currently existing techniques. When the method is applied on rabbit, it can derive high affinity antibodies with a KD value usually around tens or hundreds of picomolar range. When it is applied to alpacas, it can be used to develop a single domain antibodies more rapidly. I also showed you that by using the transgenic animals or the special strains we developed in the past, SBEAD monoclonal antibody is further facilitated to either accelerate the monoclonal antibody discovery speed, or facilitate or make the antibody screening more convenient or more accurately. So I will stop my presentation here. If the audience have any question, I would be more than happy to answer. Thank you.
Thank you, Dr. Xia. Now we will be taking some questions. The first question is, can you comment on what human germlines are closer in sequence similarity to the antibodies you obtain from the Huab rabbits?
So we only know that we don't know too much about the human rabbits. We only know that the light chain of most antibodies from these rabbits does not have the disulfide bond. In terms of other CDR region sequences, we don't know too much about them. But these antibodies, because they do not have the special disulfide bond, they can be more easily humanized.
Okay, the next question is, can you also generate polyclonal antibodies in parallel with monoclonal antibody development?
We usually use a spleen from the animal for monoclonal antibody development, so that you can still save the serum for polyclonal antibody generation. So these two processes can be run in parallel, i.e. you can get the polyclonal antibodies as well as monoclonal antibodies from the same immunized animal, either it's a rabbit or alpaca.
Okay, the next question is, can SBEAD be applied to human samples?
Yes, we tried the monoclonal antibody development in human samples, but it does require a lot of PBMC. We typically use more than 100 microliter blood samples to successfully generate monoclonal antibodies from the human samples. So I see one question here. What is the typical cost of making one antibody against the peptide antigen? So, Lexi, should you answer this question or should I answer this question?
You can go ahead.
Okay. So I would say it's around $20,000, including the animal immunization and the single B-cell cloning.
Okay, and now we have one last question. Can antibodies be developed with low amount of antigen?
Yes, it's possible. Like I mentioned, especially using the high immune rabbit, because it's typically generating more B cells for cloning. So it's possible to use much less antigen. A typical immunization of a rabbit takes about 1.5 milligram antigen. using this rapidly, it is possible to generate enough B cells with much less antigen, for example, 0.5 milligram antigen or even less. But this is not guaranteed. I mean, it could fail. I mean, we have many successful experience, but we also have experience which it did not succeed.
Okay, thank you all. Those are all the questions that we have time for, but we will definitely answer your questions individually by email. Please send your additional questions to info@appliedstemcell.com as seen on the screen. We will also be sending you a copy of today's presentation along with the recorded webinar link. Please visit our website for more information on our rabbit monoclonal antibody development and antibody humanization services. Thank you so much for attending and have a good rest of your day.